Hi everyone, and welcome to Ajua Biotech. It's so awesome to have you. Thank you for subscribing and for your comments and your likes. So in this video, we're looking at what you should be downloading, sequences that you should be downloading when you want to construct a plasmid vector for expressing a particular gene via cloning. So in my use case, this one is a plasmodium gene, but it can be any organism gene. So when we are constructing a plasmid vector to clone a segment of a particular gene using restriction enzyme-based cloning, it is crucial to select the appropriate sequence type from NCBI. The choice depends on the part of the gene you need for cloning, and it impacts how the plasmid will express the gene in your desired system. Let's have a look at the sequence options from NCBI. NCBI offers several types of sequences for genes, and each has its specific use case. Here's a breakdown of the sequence types. One type of sequence that you will find on NCBI is the genomic DNA sequence, gDNA. Here, the genomic DNA sequence includes the full sequence of the gene, along with all introns, exons, and any regulatory regions, so your promoters, enhancers, untranslated regions that are in the 3' or 5', etc. The use case for these type of sequences. These type of sequences are useful if you are working on an organism at the genome level project, such as studying intron, exon splicing, regulatory regions, or if you need a large genomic fragment that contains more than just the coding sequence. The disadvantages here, and I'm using plasmodium for a study case, is that the plasmodium genes typically have very few or no introns, so genomic DNA may not be very different from the coding sequences. So for expression, genomic sequences with introns may not be correctly spliced in systems like E. coli or mammalian cells, making them less ideal for protein expression cloning. So the conclusion then for genomic DNA sequences when you're looking at cloning a plasmid via restriction enzyme or using restriction enzyme cloning to create your plasmid is that it's not recommended for simple gene expression purposes, unless, of course, you're trying to look at analyzing non-coding and regulatory elements. The second type of sequence you will find on NCBI's database is the transcribed version of the gene, so the messenger RNA. But this messenger RNA includes both the coding sequence and the untranslated regions that you will find at the 3' and 5'. The messenger RNA or mRNA sequence represents the gene after transcription, but before translation into protein. So the use case for mRNA sequences are if you are interested in studying untranslated regions, so your 5' UTR and 3' UTR, or if you want to study post-transcriptional regulation. The disadvantages then are that mRNA contains your untranslated regions, which are typically not necessary for protein expression. There may be regulatory elements in there that are required for promoters, binding, and all that. So if you need it, then you may want the mRNA, but generally not necessary for protein expression, especially if you are focusing on the coding region for functional studies. UTRs can sometimes hinder expression, so using mRNA sequences might add unnecessary complexity. So the conclusion then is that it's not recommended for cloning unless you specifically need UTRs for regulatory or stability studies. The third type of sequence you encounter or you can find on your NCBI database is the coding sequences. This is abbreviated CDS. The coding sequence or CDS represents the portion of the gene that codes for the protein. So this excludes any introns or UTRs. It's derived from the messenger RNA, but only includes the exons that will be translated into the protein. So the use case then is that this is ideal for cloning when your primary goal is to express the plasmodium protein in a heterologous system, for example, in E. coli, in mammalian cells, or plasmodium transfection. The advantages of using the CDS is that you don't need to worry about introns, and also the CDS is concise and directly translatable into protein, and so it makes it ideal for expressing the protein of interest in recombinant systems. So in conclusion, the CDS sequence is the recommended sequence for cloning when the goal is gene expression in an experimental system, particularly for protein production. The fourth type of sequence you will encounter in the database is a gene sequence. So the gene entry typically includes the full locus, meaning it contains the entire sequence of the gene, including introns, exons, promoter regions, and sometimes flanking sequences. The use case for the gene entry is that it's similar to genomic DNA and is useful when you want to study gene regulation, such as promoter activity or genomic interactions. Disadvantages are similar to the genomic DNA in that it does contain introns, it does contain non-coding regions and flanking sequences that might not be relevant for your protein expression goals. So the conclusion is that it's not recommended for basic cloning or expression experiments. So let's look at the practical steps then. Let's use plasmodium as the example, a plasmodium gene. So if your goal is to clone a segment of a gene into a plasmid vector for gene expression, the first thing you do is download the CDS or coding sequence. The CDS gives you the exact region that translates into the protein. It avoids non-coding sequences like introns, untranslated regions, and promoter regions, which may complicate cloning and expression. It is ideal for downstream applications like expressing the gene in bacterial, mammalian, or in my case, or if you're using plasmodium cell lines. The next thing you want to do is consider any modifications that you may need. So typically you want to add restriction sites. Ensure that your primers for amplifying the CDS contain the appropriate restriction enzyme site for cloning into the vector. You should also check that it has a start codon. So you confirm that the CDS includes a start codon, and this is the nucleotide ATG. If not, you may need to add this in your primers to ensure that the sequence, the polymerase knows where to start copying. A stop codon. You may need to ensure that the CDS includes a stop codon. There are 3 versions of nucleotides that can read as a stop codon. So there's TAA, TAG, or TGA. If you are tagging the protein, for example, with a fluorescent protein, such as the green fluorescent protein, the enhanced green fluorescent protein, or a HA tag or a his tag, then you may need to modify the stop codon in your construct to allow the addition of the tag. So that brings us to the end of this video on which sequences to collect from NCBI in order to construct a plasmid vector for introducing a gene. In the next video, we're going to look at the steps for the practical side of preparing the vector and then going on to transfect the vector into your cell line of choice. I'll see you in the next video. God bless. Bye.